is Dan Freeman from Mass General Hospital in Boston, and what we're looking at now is how retinal neurons respond to electrical stimulation. We're trying to restore some functional vision to the blind using electrical stimulation with extracellular electrodes. And in particular, what we're looking at is the response of bipolar cells, retinal bipolar cells. What we found last year in a paper that came out was that bipolar cells tend to respond robustly to low stimulus frequencies, but not to higher stimulus frequencies. This is with sinusoidal stimulation. And we wanted to investigate why that is. And so we're using a computational model, a few different models, to try to answer that question. And our initial hypotheses were that perhaps it's just the passive electrical properties of the membrane, that is the resistive and capacitive properties of the membrane that are just causing this filtering, this low-pass filtering, causing the cell to respond at low frequencies but not high frequencies. An alternative hypothesis is that the cell can actually respond to a whole range of frequencies at low or high frequencies, but that the calcium channels that actually mediate synaptic release are themselves low pass. So the voltage across the membrane perhaps is modulating at very high frequencies, but the calcium channels just can't keep up, and so calcium doesn't enter the cell. So the first model that we have here is just a basic two-compartment model with a single compartment representing the terminals, the axon terminals, and another compartment representing the soma. These are connected by just a single resistor representing current flow along the axon. The extracellular stimulation is represented by this battery here, V-stim. We will modulate V-stim sinusoidally across a range of frequencies, and not surprisingly, we get this result, just a low-pass filtering. So the resistive and capacitive properties of the membrane of this very simple model result in this low-pass shape. But importantly, this cutoff frequency is very high. This is about 900 hertz. And so we know bipolar cells don't even respond to 100 hertz. They respond more to about 20 hertz. So this is a relatively high cutoff frequency, but it's a simple model. So we said, why don't we create a more morphologically realistic model? And so we took this morphological reconstruction of a bipolar cell, and we created this more complex model, a multi-compartment model, made up of many compartments, each of which represents some patch of the membrane. And again, you have resistance along the intracellular space, representing resistance to current flow. In this case, our stimulus is represented not by just a single battery, but by some point source positioned about 40 microns from the axon terminals. The voltage elicited from this point source falls off inversely with space, represented by this equation here. And what we're going to do is just use this equation and basically represent that with an array of batteries. So each battery applied extracellularly is going to have some value, and that's going to fall off with space, so with distance from this point source electrode. We're going to, again, modulate V sinusoidally and measure the membrane potential in the terminals. And we're just going to measure the frequency response. And not surprisingly, again, we got a low-pass filtering. And it looks very similar to this simple model that we saw earlier. Cutoff frequency is a little different, around 700 hertz, but that's still very high. And we said, well, why don't we tweak the parameters a little bit? Some bipolar cells are bigger than others, bigger soma, bigger axons, longer axons. So what we found is if we start tweaking the axonal resistance over some large range of values, the cutoff frequency will vary, but it still remains relatively high. It remains above 100 hertz for any axonal resistance we choose. So what this tells us is that the passive electrical properties of the membrane cause low-pass filtering, but we don't think that's why bipolar cells respond to low frequencies in terms of their synaptic output. We think that perhaps it's the calcium channels. But we can actually test that with another computational model. Calcium channels have been characterized with the Hodgkin-Huxley formalism represented by these equations, both L-type calcium channels and T-type calcium channels. These are the channels that are responsible for synaptic release from bipolar cell terminals. So all we're going to do in this case is just modulate the voltage across the calcium channels and measure the resulting current. And you're going to get some sort of filtering because of the voltage-gated properties of these channels. So what we found was we measured the frequency response and just plotted the cutoff frequency for each of these channels as a function of stimulus amplitude. Because these channels are nonlinear, the cutoff frequency will vary with stimulus amplitude, but generally what we found was that the cutoff frequencies are pretty low, both for L-type and T-type channels. So the L-type, they actually get down in the range of about 50 hertz. In T-type channel is always very low. It's always well below 100 hertz, so around 25 hertz, the highest that it gets. So what this suggests is that when you stimulate a bipolar cell with electrical stimulation, it may be able to respond to very high frequencies. That is, its membrane potential may be fluctuating at 100 hertz or 200 hertz, but it can't produce synaptic output because those calcium channels that mediate synaptic release just can't respond at those very high frequencies. So that's the basic conclusion.
Thank you very much.
Yep.